Hello everyone, welcome to part 3 of the video series. So this video is about DNA copy number alteration and GC bias. So both of these topics are extremely important because we are going to encounter copy number profile many times, and GC bias you will hear about it many times. Now when I was starting off my PhD, I actually looked for the GC bias content in YouTube and in different places, but I didn't really get something simplified which would satisfy my needs. So I will try to satisfy your needs in that regard. One thing I also want to mention here is that before watching this video, you should definitely go through part 2, because part 3 is sort of dependent on part 2, because in part 2 we talk about sequencing, and this particular tutorial also talks about sequencing, and I will sort of guess that you already know that stuff, so I will not talk about that. So let's get started. So first of all, if we do whole genome sequencing, I've talked about this in a previous video. If we do whole genome sequencing, then approximately the reads that we are going to get should come uniformly across the whole genome. So for example, I have divided this genome into 4 parts, 1, 2, 3, 4. And in each part, if you count the number of reads, they are approximately equal. Now, that should be the case ideally, but often that doesn't happen. Let me now talk about why it doesn't happen. So sometimes you can see that in some varied bins of the human genome. So in human genome, there are actually some genomic regions which are actually not very well defined, or sequencing doesn't work really well on those regions. So if we just exclude those, if we just include the normal regions, which is most of the regions in the human genome, then in some regions you can see this kind of thing where you have barely any read, and in some places you can see like 2x or 3x number of normal read count. So for example, here we have approximately 8 reads, but here we have approximately 15 to 16 reads, which is like double. So why does this kind of deletion or amplification happen, or why does this kind of doubling actually happen during sequencing? So if we talk about that, then we talk about the copy number alteration. So first of all, this is a coverage profile. So when you define the entire genome as counts of equal sized bins, so for example, suppose each of these bins can be 1 megabase in size, right? 1 megabase or maybe 100 kilobase or whatever is convenient for your analysis. So if you actually take the count of reads for each of these bins, then that is basically a coverage profile. So it's just a count of reads. So we can have some copy number alteration in human body. For example, if we look at this pair of chromosome, as we talked about in part 1 of the lecture series, that we have chromosome pairs, right? We have 23 chromosome pairs. So in some chromosome, if we have some region which gets duplicated, right? So you can see it got duplicated right here. Then we can have some amplification, for example, like this. Or if there is some deletion of some part, for example, this part got deleted here, we can have some deletion, which is shown right here. Now, what I've shown right here is an example of heterozygous duplication or heterozygous deletion. It is because both of the chromosomes did not actually encounter this phenomenon, right? Only one chromosome encountered this. But if both chromosomes encounter duplication or deletion, then it is called homozygous alteration. So first of all, as I mentioned before, it can happen at chromosome level. So this is basically large scale copy number alteration, and you are going to see a lot of amplification, deletion, or duplication in such cases. But it can also happen at a much smaller scale, like 100 kilobase region, which is a very small region in the human genome. It can also happen like that. And this is very, very important if you are doing cancer research, because it is one of the hallmarks of cancer. If you have cancer or if you have some physiological stress in your body, then this kind of deletion duplication can happen in the DNA of human cells. So as I mentioned before, homozygous and heterozygous alteration, so this is mentioned here. And if you want to detect the copy number altered segments in your human body, like which segments in your human body are actually copy number altered, there are actually tools available. For example, CNV kit, GATK, GCNV, etc. And finally, for different diseases, for example, if you have like liver cancer or if you have breast cancer, there are some copy number signatures. So there are some predefined bins and predefined pattern of copy number which can be used to classify that cancer type, identify that cancer type. So it's a very important property in bioinformatics, the copy number profile. So as I mentioned before, the phenomena that I talked about right here, amplification or gain or deletion or loss, main reason is copy number alteration, but there is an artificial effect which is also on the run. And this can hamper your analysis. This can destroy you completely. So that thing is called GC bias. So I will talk about GC bias right now. So imagine that this is a normal genome, and in this normal genome, you have 1, 2, 3, 4, 5, 6 bins, right? 6 bins. So each of these bins, let's say they are of size, how much? 1 megabase. They can be of 1 megabase, 100 kilobase, anything you want. So let's assume that they are 1 megabase. And obviously, you can count the number of reads. You can take the read counts from each of these bins, right? So these are the read counts, 200, 100, 205, etc. So you can see that there is quite a lot of variation in these counts, right? Now you may be wondering that, okay, most likely this is because of copy number alteration. But wait, there is another artificial thing going on here. Let me talk about that now. So what is GC content? So let's say this is a 1 megabase size bin, right? This is a 1 megabase size bin. That means there are 10 to the power 6 bases in this bin. There are 10 to the power 6 characters of ATCG in this bin. And among those characters, 32% are actually G or C. Okay. So if you count the total number of G bases and the total number of C bases, their sum, the count of their sum is 32% of the total string size 10 to the power 6. Right. So that is called GC content. And you can see different GC contents here, right? 47%, 67%, 50%, etc. Okay. So you can see that the count sort of correlates with the GC content. Let me actually show you how it correlates. If you have a very normal GC content like 50%, so half of the bases are G and C, which is basically what we expect, right? Because GC is 2 bases and AT is 2 different bases. So in that case, the read count is around 205, 200, etc. 47%, 200, 50%, 205. So this black segment and this black segment, they are like normal GC content they have. But if you look at the red segments, so these red segments have a relatively high GC content, right? 67% and 70%. So you can see that their read counts are actually much smaller compared to the normal black segments, right? Also, you can see 2 yellow segments here where GC content is very low, like 30% and 32%. In those cases, you can also see the same trend. You can also see 87 and 95, right? So which is not really, I would say, even close to 200 and 205. So why does this thing happen? So let me explain this very briefly. So first of all, obviously during WGS or whole genome sequencing, as I mentioned before, if there is no copy number alteration, the reads are supposed to be uniformly distributed across the whole genome. But the count can be affected by the availability of GNC bases in different regions, so which is called GC content. Now the issue is you can see less reads are derived from GC rich or GC poor region. So for example, in the GC rich red region or GC poor yellow region, the count is less, right? This is called GC bias. Now, why does this happen? The main contributor of GC bias is the PCR step of sequencing. So I talked about the PCR step in previous video. So PCR is the polymerase chain reaction, which is used to amplify input DNA, right? Because we obviously give a small amount of DNA, amplify it in order to sequence it properly. Now, this amplification efficiency depends on the GC content of different region. So if some region of the DNA has high GC or very low GC, then that portion is not amplified that much. So understand that this GC bias, it is actually an artificial bias. It is not something biological, and we definitely have to account for it. That is the important thing. Let me see if I can write something here. So there are actually tools. For example, GC Fix. This is a tool that I've made in order to correct for GC bias. Then we have Griffin. We have a particular tool called LOES. Then we have GC Paragon. Okay, so we have all these tools. We have all these tools that can actually correct the GC bias for you. So if you are doing copy number analysis, if you are doing copy number alteration analysis, you should definitely correct GC bias first, because if you do not do that, then you are going to find copy number altered regions which are not actually altered and which are altered simply because of the GC bias. Okay, that is the important thing. So okay, let me cross this out. So one last thing, so you can see a GC bias curve right here, right? So GC content is in the x-axis and the mean coverage is the read count basically, the mean count is on the y-axis. So you can see that it's sort of a bell-shaped curve. So if you actually like count these things, count the mean coverage and calculate the GC content, taking a sample, taking any sample, then you are going to see this kind of curve generating, right? It's a bell-shaped curve. So this is DC bias. So that's it for this tutorial. So in the next tutorial, I'm going to talk about methylation in the human genome and epigenetics. So epigenetics is the real hot topic now because that is part of developmental biology. So I will talk about that in the next tutorial. Thanks everyone. Hope to see you in the next tutorial.